All right. So let's get this review session started. Some things I want to mention at the very start. Um, if you notice, well, maybe not, but just earlier this morning, I made all 4 tests available for you to repeat as many times as you want. So the bottom of course content here, you'll see test 1, 2, 3, and 4 repeatable. And these are just the tests that you can try as many times as you like. I think this is a good place to start, like really reviewing, in that if I ask something on the test, there's a high chance I might ask it again on the final exam. And by redoing these tests, hopefully you can kind of look at, okay, what do I remember? What do I not remember? Right? So there's that. Also, the final exam, as you can see here, it's hidden to you. I have my rough copy done. It's roughly what, 85-ish questions, but the vast majority are multiple choice. So there might only be like 5, if that, short answer questions. So a lot of multiple choice. Hopefully time won't be an issue because you should have, or you will have 2.5 hours to take this. Again, the final exam is available Thursday and Friday. Please give yourself time. Don't wait till the last minute on Friday to do it in case technical difficulties do occur. That being said, I'm sure people will be delighted to hear this. I won't be using ProctorU for the final, so you don't have to worry about those technical difficulties. And the test, therefore, I will allow you to use your notes for the test. That being said, please still study, because if you're trying to look through your notes for every single answer, you might have a hard time finishing. So still study, be prepared, but yes, you can use your notes on the test. Okay, so with that out of the way, are there questions that I can answer for anybody? Anybody still have wondering about anything? Want me to go into detail on something? Anything at all? Feel free to fire away with any and all questions. How much is our final worth again? I think it's 22%. And that reminds me, I didn't do it yet. I need to put up an Excel file that will walk you through how to basically determine what you need to get on the final to get a certain grade in class. So I made an Excel sheet to do that. So I can put that available. Can I go over March 9th, question 1? Sure. So let me open up March 9th. Okay, so math question, no problem. Not too sure if you'll test us on it since it wasn't on the exam. If it wasn't on the exam, there's a good chance that I won't ask you it. So the way I'm trying to make my finals is that if you saw something on test 1, 2, 3, and 4, it'll probably be on the final. But if I didn't show it to you, then I probably won't ask you a question on that. So that being said, I probably won't ask you a question exactly like this, but we can go over it here just super quickly. Actually, let me just stop the sharing. Let me let me actually check something. Dun, 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 dun. Right. So yeah, I will say, yeah, if I didn't ask about it in test 1, 2, 3, and 4, like a math question like this, then don't worry about it. Save yourself a little bit of time. I can still go over it if you want. Yeah, that's what I figured. No questions. What do people want to know? Still confused about something? Maybe a question on the test you didn't quite get. And while you think about that and look over, by the way, your test, you should be able to look at your tests. If you can't, let me know. But while you're thinking about that, let me share question that I did get via email. So the email question was, what's the difference between PP1 in the muscle and in the liver? So if you remember PP1, or phosphoprotein phosphatase 1, it is one of these molecules that helps control glycogen, the production or breakdown of glycogen. And PP1 is a phosphatase. So a phosphatase generally removes a phosphate group. And just give me 1 sec. My fan is making a super loud noise. It's really distracting for me. All right. And PP1 is controlled a little differently than in the muscle and the liver. And so in the muscle, PP1 is bound to this GM subunit, right? And this GM is what's connected to glycogen. Now, if PP1 is connected to GM, then it is active. It will do its reactions here, but not that strongly. If GM gets one phosphate group, then PP1 becomes activated. And we can get a phosphate via insulin, right? So insulin comes in, says we have a lot of glucose in our blood. One of the things insulin does is that it will turn on a kinase that will add a phosphate group to this GM subunit. Once this GM subunit is phosphorylated, PP1 is activated, which will stop glycogen phosphorylase. It will stop phosphorylase kinase. Therefore, it will stop the breakdown of glycogen. However, if GM gets a second phospho group, and then PP1 will stop interacting with GM, and PP1 will no longer be active, leading to the breakdown of glycogen. So that is how in the muscle PP1 is regulated. It has this GM subunit, and GM is being phosphorylated, and the phosphorylation signal is regulating activity. No phosphates, somewhat active. 1 phosphate, really active. 2 phosphates, not active. And then if we go to the liver, the way PP1 in the liver is activated is that you don't have like a GM here, right? GM is for the muscle only. And PP1 doesn't have the same phosphorylation scheme here, right? The way PP1 turns on in the liver is that you bind to the enzyme phosphorylase A. Again, this is a little bit of a blast from the past because this was, back in February, I think. Phosphorylase has two different forms, the alpha and the beta form. Alpha really wants to break down glycogen, so it's usually on. The beta form is usually off. When glycogen phosphorylase A is in the R form, which is here, you want to break down glycogen. So what it also does is that it will bind PP1 to itself. Once PP1 is bound, it can no longer dephosphorylate glycogen phosphorylase or phosphorylase kinase, right? So basically, if you turn on phosphorylase A, this is turned on, you more or less take PP1 out of the cell because you grab onto it. However, if you go to the T state, PP1 will then chop off the phosphoryl groups of phosphorylase alpha, switching you to the inactive form, right? So the big takeaway of PP1 regulation in the muscle versus the liver, in the muscle, you're bound to GM and you are controlled via phosphorylation. In the liver, you are controlled by phosphorylase alpha. If you're in the R state, PP1 is sequestered and not allowed to do anything. If you're in the T state, PP1 is released, and then you inactivate phosphorylase alpha. So that's the answer to that question. What other questions do people have? Be about anything and everything. Well, here's another question from the internet. While you look over your notes, look over your tests. So a question, another question I got via email is to answer this question from test 2. A person has a mutation in complex 3, so you cannot do the Q cycle. Would you or could you still produce energy using the electron transport chain? Please explain your answer. All right, so if you remember complex 3, what complex 3 does is that it takes the carrier from complex 1 and complex 2, the electron carrier, and it takes those electrons from coenzyme Q and gives them to cytochrome C. And it does that in 2 steps, which is called the Q cycle. When it does that, it pumps hydrogens against the gradient. So, right. So this is complex 1.
complex 2, complex 3, and then complex 4. Right, so both complex 1 and complex 2 send coenzyme Q. Complex 1 will pump hydrogens out when it does that. Complex 2 doesn't. Complex 3 sends cytochrome C to complex 4 and pumps out hydrogens when it does that. Then complex 4 allows hydrogens to go through naturally, and we make ATP, electron transport chain. And my reasoning for that question was, if complex 3 is knocked out, you would still have protons being pumped from complex 1. So it is possible to still maintain somewhat of a proton gradient to allow ATP to be made in complex 4. If you wanted to make the argument that you would not produce energy because after a while, your coenzyme Q would never be regenerated and you would run out of coenzyme Q to give electrons to, therefore stopping complex 1, I would be okay with that as well, because that makes logical sense. This is kind of looking at how well you understood how all the complexes work together. And if you knock one out, what's the consequences? All right, so those were like the two main answers that I would accept for full credit. And I think for this one, based on how you answered it, I gave people some partial credit here and there. But that was a question that was asked online. And along with that, a question I got was, are electrons accepted by complex 1 higher energy than those accepted by complex 2? So are electrons accepted by complex 1 higher energy than electrons accepted by complex 2? And for that, I would say the electrons themselves have the same energy. But the redox reaction you're doing are different energies. So the redox reaction in complex 1 is higher energy than the redox reaction in complex 2. And complex 2 is so low energy that you cannot pump protons across the gradient. You can only send coenzyme Q. Complex 1, you can pump protons across the gradient with that energy. And I had one more question online that had to do with test 4. So let me get to it. Let me stop share so I can move over to test 4. And let me pull up the relevant PowerPoint. Right. Yep, so I'll get to those questions that were just put up in chat in 1 second. Let me get to this one that was online. So the question was, could you explain why the answers for select all that are true about splicing are shown here? First off, let me talk about the false one occurs in the cytoplasm. And I guess, so this is test 4. So in the exam, I would probably be a little more specific when I say splicing. This is mRNA splicing, right? So this occurs in the nucleus. Right after you're making your mRNA from your DNA, right? Splicing is going to happen. So that's why that occurs in the cytosol. It's not true. But let's look at the other one. Results from 2' hydroxyl attack on the exon, attacking the 5' splice site, is completed with the 3' hydroxyl on the newly cut exon attacking the 5' terminal phosphate on the next exon. So let's just review that. And where did it go? I just had it up. Okay. So basically here is the reaction again. Remember, you have an invariant adenosine in your intron. And this is RNA. So RNA, we have a 2' hydroxyl. The first thing that happens is this 2' hydroxyl attacks the 5' splice site. So that was the first true statement. And we get this lariat structure. Then on the exon you just created, you have a 3' hydroxyl hanging out because you broke that phosphate bond. So the 3' hydroxyl goes and attacks exon 2, and you have excised your intron. So that's why on test 4, those were the 2 true statements. Let's see the other messages I got now. For the question on test 4 on the nucleotide cap eukaryotic mRNA, aren't there 2 different nucleotide capping each side, a G on one end and a poly A tail on the other end? Okay, so those are 2 different like terminologies. The cap is at the 5' end, and that's guanosine. The tail is at the 3' end, and that's the A nucleotide. So usually that's how people will talk about those. They'll talk about the mRNA cap and the mRNA tail, right? So that's why the answer for what was the cap is M7 guanine. Can you do question 13 from test 1? Sure. I will say, if you ask me specific questions about the test, could you do me a favor and tell me what the question is? Because everybody has a different order of questions. And I can look it up on a person-to-person basis. But it'll just speed it up a little bit if you could just tell me like, what is the, like, at least the beginning of the question so I can copy and paste or control F it. Let's see, test 1. Where are you, test 1? Is that hidden from me? Let's see here. Sorry, give me 1 second here. It starts with compound K as the ultimate product. So let's take a look here. I think I know what question it is. Yeah. This fun question. All right. So here on question 10 for me, this is glycolysis in a nutshell. I'm just taking away all the words of glycolysis to see if you can understand, the different types of way glycolysis is regulated. So here we're making the molecule K, and it's linear.
It has 10 different steps, just like glycolysis. So let's see what this description says. K inhibits enzyme 5. So K will go and inhibit enzyme 5. Enzyme 5 is therefore under what type of control? Right. So covalent modification means you add like a phosphate onto it. That's the type of control we were talking about earlier with PP1. So this isn't covalent modification. Genetic control is when you either increase or decrease the amount of enzyme by increasing or decreasing the amount of like mRNA or the amount of transcription. Here, we're not talking about influencing mRNA production, so it's not genetic. So this type of activity or control is called allosteric. And allosteric means that, binding a molecule either speeds up or slows down a reaction. That's what allosteric control is. So here we bind K, enzyme 5 binds K, and it slows down. It inhibits it. So that's allosteric control. All right, so moving on. If enzyme 3 becomes activated in response to the activity of pKa, only when compound A is present, then enzyme 3 is under what type of control? Okay, so product A turns on pKa. pKa adds phosphates to groups, and so it will add a phosphate to enzyme 3. As I mentioned, this is covalent modification, covalent control. So the other ones are out already because it's not genetic. But let's continue. If enzyme 2 is only expressed in the presence of a high concentration of A, then 2 is under what control? So here we're talking about levels of the protein, where A will directly control how much 2 is being produced. Since we're talking about production of a protein, that is genetic control. If all of these are true, high concentrations of what will decrease the flux? And so here you only had 2 possibilities, K and A. You can see A turns on 2 and 3. So that's not decreasing flux. But high levels of K inhibits enzyme 5. Therefore, if enzyme 5 is inhibited, you will not continue through this chain. You're decreasing flux. This type of question is like a question, those of you who are thinking of like taking the MCAT, they love throwing generic garbage at you like this to see how well you understand these terms. Because if you can puzzle out like a question like this, I can basically give you any new metabolic pathway that you've never studied before, and you can at least pick it up relatively fast because you get all the terminology. So that's kind of what that question is testing on, the terminology of our metabolic pathways. All right. What else do people want to know? In regards to select all that apply, question 3 during test, gluconeogenesis living during starvation is incorrect. Can you help me with that? Sure. So let me see if I can pull up the slide for that. Because the reason why that happens might not be obvious. I think it's this slide. Huh. Let me just double check test 3. Which of the following happens in starvation? Kidneys generate gluconeogenesis. Okay. So this is what, okay. So what I'm gonna do is, I think there's actually 2 correct answers for that question. It depends when you define starvation, and I guess I was thinking long-term, and then I just checked what the actual PowerPoint slide said, and it has a different term. So let me pull up the PowerPoint. All right. So in the actual PowerPoint, gluconeogenesis increased, supplying 96% of glucose reduced by the liver after 40 hours of fast. So that would say during starvation, gluconeogenesis increases in the liver. However, I guess I was thinking when I originally wrote that question, long-term starvation, gluconeogenesis will start to taper off in the liver. And what's going to happen is that you're really going to get your glucose from your fats and triacylglycerols rather than the liver pathway. So for question 3, let me just share that question with everybody. Which of the following occurs during starvation? What I'll do is I'll go back for people and accept either 1, 2, and 3, or 1, 2, 3, and 4, because I was not specific enough when I said starvation, because it changes depending on how long you starve. So let me make a note of that. And so you might see your grade on test 3 increase a little bit because of that. All right. Yeah, kidneys are generating glucose. Yeah, but it's short term, yes. Long term, they start to taper off a little bit. And the liver mainly focuses on ketone bodies at that point. I think the transition to long term is going to be I think it's even longer than 48 hours. I actually don't know. Because long-term starvation, um, well, first off, I haven't looked at it that much. Just going off what's in your book, they have the marker at like 40 days. You're really just using ketone bodies at that point. So I would say probably like, you know, after a week or two, you are gonna start making different enzymes. But I don't actually know the exact time frame. What else? Any questions about anything at all? Anything on the test you want me to go over? Anything on the slide you still didn't quite get? Let me see. Glucagon and its control over muscle metabolism. Yeah, I think that's, well, that's not on the same slide as the other one. Because I know there's a nice little table that goes over what happens when glucagon and all the other hormones are released. So let me see if I can't find that really quick. Yep, here it is. And I think this one's a little tricky because yeah, it's just a little tricky. So glucagon does not have a direct impact on the muscle. Your muscle does not have any glucagon receptors. So if I release glucagon into the body, it does not directly respond. However, what it will do is that it will go to the liver and start releasing glucose from the liver and into the bloodstream, which can make the muscle do more glycolysis at that point. So there's no direct effect by glucagon, but there are indirect effects by glucagon. And then the fat, it just makes fatty acids be released.
Yeah. So that's why the question I'm guessing is metabolism in the muscle is controlled hormonally by and glucagon, insulin, and epinephrine. So yeah, no direct effect, but there is indirect effects. And I think I want to say during that lecture I might have specifically said something like that, just because I knew I was going to ask that question on the test. So I really wanted to point that one out. So yes, indirectly, it makes, well, glucagon makes the liver release glucose, which indirectly makes the muscle uptake that glucose and start metabolizing glucose. Yeah, so yeah, that's why that question, that's why I went on my way on that question when we went off that slide to try and emphasize that, because I know that question's tricky. It's, yeah, because it's indirect control at that point. All right, so another question I got is from test 2. So let me pull that one up. What are the 3 drawbacks of aerobic metabolism? That is, what's bad about breathing oxygen? And there's a lot. One, if you don't have oxygen, you are damaged forever. So yeah, you need oxygen, irreversible damage. 2, you produce reactive oxygen species. Those can affect a lot of things. And so if you said anything along with reactive oxygen species, such as DNA damage, lipid damage, enzyme damage, mitochondria damage, is the reason why we probably age. I was looking for those type of things. So basically, why is breathing oxygen bad? Damages your DNA, damages your proteins, damages your lipids, damages your mitochondria. So yeah, that's the main things. Just seeing if you could point any of those out. I want to say for a lot of these, if when looking back on your test, if you don't see an answer there, it's probably because I forgot to put it up. So just let me know, and I'm gonna double check that right now. Whoops, you don't want to see that. Let me stop sharing before I click buttons. Feel free to ask more questions while I make sure you can see all the tests. All right. Anyone else? Or are we feeling good at this point? Because if you don't have any pressing questions, what I would say is look over your notes, organize your notes. That's going to be a big thing because it is open notes. Go and retry the tests, right? So you can focus on like what is your weaknesses still, what don't you quite remember. And if you have questions popping up, always feel free to email me. let me know and I'll answer those as best as I can. Last call. Well, if there isn't any, like I said, I'm pretty much available more or less all day until you're until summer semester starts, at least, because I basically have all the finals written. So until I have to start grade them, I don't have much going on. So if you do have questions, you want to have another meeting, let me know. We can set that up. Otherwise, good luck studying. Make sure to not stress yourself out. If you feel yourself stressing out, go and pet a dog, pet a cat. Science has shown that releases endorphins and it makes you feel good. Exercise, eat well, sleep, especially before the exam. Do not like cram all night. Well, for you, you don't have to do that because you have 48 hours to do it. Just make sure you get a full, like, at least like 7 hours. A rested brain is a well-thinking brain. Otherwise, take care, everybody. Good luck and have a good summer.